Welcome to the Deep Dive. Today we're doing something a little different. We're giving you a rapid, highly filtered injection of the most important protocols defining oncology research right now.
Exactly. We're getting into the nitty-gritty.
We are taking a deep dive into the absolute cutting edge of cancer drug resistance. These are the critical, practical lab and computational methods that, if you're a medical student or a young professional, you just have to master.
Well, you do, because the enemy here it's just incredibly resourceful. And the sources we've compiled lay out the modern toolbox that's been built to fight it.
And we're not just talking about observing things anymore.
No. The whole point is to identify resistance mechanisms at the deepest level possible. So molecular, cellular, and functional.
And these are not old-school methods. We're talking about advanced cell culture, microfluidics, tumor microenvironment systems, all integrated with some really high-powered analytics.
That's it. It covers everything from functional assessments in vitro and in vivo to really sophisticated single-cell work like mass cytometry. And all of that data feeds into these massive computational pipelines.
The ones that handle all the abonomics, data genomic, epigenomic,
transcriptomic, metabolomic, all of it. So our mission for you really is to synthesize all these state-of-the-art methods into a clear conceptual framework.
Okay, let's unpack this. And let's start where cancer research has, for a long time, had its biggest bottleneck: building better models.
Right, because the simple two-dimensional Petri dish culture, it just fails spectacularly at recreating the complex environment of an actual tumor.
If your model doesn't reflect reality, your results won't either.
That's the starting point, which is why three-dimensional or 3D biomimetic systems are now, well, they're essential. They're designed to mimic the architecture and the mechanics of real tissue much more closely.
And a big conceptual breakthrough here is moving away from old scaffolding materials, things like matrigel or collagen.
Yes, which are animal-derived and chemically all over the place. They introduce huge variability, not to mention the ethical concerns.
So what's the alternative?
The leading method now focuses on materials like alginate. It's a natural, biocompatible hydrogel that's actually derived from seaweed.
Seaweed, so it's a defined non-animal scaffold.
Exactly, which gives researchers unprecedented control over the environment. No more black boxes.
But even a 3D scaffold has a limitation, right? Once the tissue gets thick, oxygen and nutrients can't get to the center, and the culture just dies.
That's the classic diffusion problem, and you overcome it by adding flow, continuous flow, or perfusion, using systems like a 3D perfused bioreactor.
And that's the conceptual game changer, the flow.
It is, because it actively maintains cell viability and phenotype. It just dramatically increases nutrient and oxygen transfer deep inside the tissue.
So perfusion is what allows for the long-term experiments you'd need to see acquired drug resistance, not just initial sensitivity.
Precisely. You can sustain these cultures for up to 28 days. That's long enough for the tumor cells to start showing the kind of resistance mechanisms you see in the clinic. The flow is very carefully managed to keep the whole thing viable.
Okay, let's make this practical. We have a workflow here for patient-derived tissue from ER-positive breast cancer. Walk us through it. How do you go from a tumor sample to a drug testing platform?
So this is where it gets really powerful. This is personalized medicine in action. After surgery, the tumor sample is processed.
Mechanically chunked up.
Mechanically, and also through a long enzymatic digestion. It's a delicate process. You need to carefully break down the bulk tissue while preserving these small, viable breast cancer tissue microstructures.
So you're not dealing with just individual cells. You have these little intact tissue fragments.
You got it. And these fragments are immediately encapsulated in that alginate hydrogel we mentioned. Then they're dynamically cultured.
What does dynamically cultured mean here?
It just means they're usually on an orbital shaker, something to provide gentle movement and mimic physiological flow. And this is all done while being challenged with drugs like tamoxifen or fulvestrant for about 2 weeks.
And the final readout isn't just counting dead cells.
No, far from it. We're looking for hard molecular evidence. The microstructures are retrieved, and drug response is assessed using RT-qPCR to analyze gene expression.
Looking for key genes.
Right, like ER-alpha and its downstream targets. And this molecular analysis gives you an ultra-sensitive quantitative score of whether the therapy is working or failing.
Okay, so that takes us from a bulk tissue model to modeling something even more intricate.
Yeah.
The tumor microenvironment, the TME.
Right, because the goal is to understand how non-cancer cells, so immune cells, fibroblasts, stem cells, how they help the tumor resist therapy. And for that, you need spatial control.
You need to study how they talk to each other without them all being mixed up in a soup.
Exactly. And that brings us to microfluidic co-culture systems.
So how do you manage that separation while still allowing communication?
A common setup uses a two-layer device. You'll have a channel layer on top of a porous hydrogel slab, maybe agarose. You could put triple-negative breast cancer cells in one channel and, say, adipose-derived stem cells in a parallel channel.
And the agarose slab is like a screen door between them.
Perfect analogy. It separates them physically but allows passive diffusion of all the biomolecules, growth factors, cytokines, all the signaling molecules between the cell types. They're talking to each other.
And I'm guessing this system also uses continuous flow.
It does. Just like the bioreactors, it allows for continuous infusion of fresh media, which maintains a stable environment. It's crucial.
This level of control is so important because the tumor isn't a static target. It's always evolving. So if we had these great in vitro models, how do we monitor what's happening in the patient in real time?
And that's the role of the liquid biopsy. It's minimally invasive and gives us this continuous real-time window into tumor evolution.
And for that, we're looking for two main things, right? CTCs and ctDNA.
Correct. Circulating tumor cells and circulating tumor DNA. And they tell us fundamentally different things.
Oh, so?
Well, ctDNA is mainly shed from dead or dying cells, so it gives you information about the tumor's cumulative genetics mutations, copy number changes.
Okay, the genetic blueprint.
Right. But CTCs, the circulating tumor cells, they are intact living cells. They're much rarer, but they provide critical single-cell biology information. You can see phenotype, protein expression, things like that.
And the sources describe a very efficient way to get both at the same time from one blood sample.
It's all about maximizing what you can get. The workflow uses a concurrent isolation protocol. It usually starts with physical separation, like filtration, to capture the larger CTCs.
And then you go back for the DNA.
Then from the remaining plasma and what's on the filter, you perform DNA extraction to get the cell-free ctDNA. This way you get both the genomic and the cellular readout from one patient sample. It's incredibly efficient.
Okay, so liquid biopsy shows us the tumor's evolution. But how do we exploit the vulnerabilities that this evolution creates? This brings us to a really elegant concept, synthetic lethality.
Synthetic lethality, or SL. The principle is actually over a century old. It's the idea that two things that are harmless on their own become lethal when you combine them.
And in cancer, that means a gene that's already mutated in the tumor.
Right. Let's say gene A is lost in the tumor, creating a dependency. The tumor cell is fine with that, but then you design a drug, anti-B, to inhibit the compensatory mechanism, gene B. The tumor can handle losing A or losing B.
But not both at the same time.
Exactly. The combined loss of A and B kills it.
So to find these pairs, the workhorse method is a viability assay to determine the IC50.
The standard is the MTT assay, usually done in a high-throughput format. And the mechanism is really cool. The MTT solution is taken up by living cells, and their active mitochondria convert it into these purple formazan crystals.
So more purple means more living, metabolically active cells.
Exactly. Researchers dissolve those crystals in a solvent like DMSO and then measure the purple intensity with the spectrophotometer. And if you see a big difference in the IC50 of that anti-B drug between your A mutant cells and your normal wild-type cells...
That's your synthetic lethal interaction. That's the therapeutic window.
That's your window.
But viability is just a snapshot. How do you confirm that you're actually killing the cells for good, not just slowing them down?
That's where the clonogenic assay comes in. It is absolutely vital. It assesses the cell's long-term reproductive ability, which is the fundamental measure of true survival.
You're seeing if a single cell can grow into a whole colony.
Right. You plate single cells and wait weeks for them to form colonies of at least 50 cells. If a drug looks great in the MTT screen but fails the clonogenic assay, you know it's not a true killer, it's just cytostatic.
Okay, let's zoom out. From specific vulnerabilities to these massive data sets, polygenomic analysis, how do computational workflows even begin to integrate 5 or 6 different types of omics data?
The power really is the integration. These pipelines ingest everything. mRNA, miRNA, DNA methylation, SNPs, CNAs, you name it, and they correlate all of it back to a drug resistance phenotype, like the LC50.
And this happens in 2 phases. What's phase 1?
Phase 1 is the high-level association. It's using linear models to find individual features, say a specific SNP, that statistically correlate with the drug resistance level. It's a hypothesis generation at a massive scale.
Just finding the clues.
Finding all the potential clues. Then phase 2 is the intellectual leap, moving from just association to defining a regulatory mechanism.
This is where you connect the dots.
Precisely. Phase 2 connects those peripheral features back to gene expression. For example, you might look for a miRNA that not only correlates with drug resistance, but also has a strong negative correlation to an mRNA.
Which suggests it's repressing that gene.
It instantly suggests a relationship of repression. And that gives you a testable, mechanistic hypothesis about how the tumor is silencing one of its vulnerabilities.
That systematic approach is crucial. And speaking of systematic, let's talk about the modern gold standard for functional screening. CRISPR-Cas9 screens.
Yes, pooled CRISPR screens. They let you perturb thousands of genes all at once. The basic method relies on infecting cells with a lentivirus that carries both the Cas9 nucleus and a specific guide RNA.
And this setup has to be incredibly precise to make sure you only hit one gene per cell.
Oh, absolutely. Researchers aim for a very low multiplicity of infection, or MOI, around 0.3. Statistically, that ensures the vast majority of cells get only one unique guide RNA, so one gene perturbation per cell.
Once the cells are perturbed, the drug dosing logic is really clever. You're screening for two different outcomes in one experiment.
That's right. You treat the cells at two distinct doses. A low dose, LD20, where 80% of cells normally survive. If a gene knockout causes hypersensitivity, that cell dies and its guide RNA disappears. That's negative selection.
And the other dose.
Then you use a high dose, LD80, where only 20% would normally survive. If a gene knockout confers resistance, that cell lives, and its guide RNA becomes enriched. That's positive selection.
And then computational tools sort through all the sequencing data to find those winners and losers.
Correct. Tools like ATAC-seq and drug-Z align all the guide RNA sequences and statistically identify the gene-drug interactions based on which guides are enriched or depleted.
Now let's talk about the layer that controls all of this. Epigenetics. If resistance involves turning whole pathways on or off, you need to map the chromatin. And that's where ATAC-seq comes in.
ATAC-seq, yeah. Assay for transposase-accessible chromatin with sequencing. It's the key technique for dissecting how chromatin structure changes during drug resistance. It uses an enzyme, TN5 transposase, which acts a bit like a molecular probe.
How does it work?
It preferentially fragments DNA only where the chromatin is open and accessible to transcription factors. So you're basically mapping all the open doors in the genome where transcription could be happening.
But just because a door is open doesn't mean you know which room it leads to. An open region of chromatin doesn't tell you which gene it's regulating.
Exactly. Accessibility is only half the story. That's why ATAC-seq results have to be integrated with other data. You combine it with RNA sequencing, which tells you what's actually being transcribed, and, most powerfully, with high-C data.
What does high-C add to the picture?
High-C is vital because it reveals the social network of the DNA. It maps the 3D chromatin architecture. It shows you which distant regulatory elements, like enhancers, are physically looping in to talk to and control a gene's promoter.
So it connects the open door to the right room.
It does. This integration lets you accurately assign regulatory regions to their target genes, giving you a complete mechanistic picture.
Okay, let's wrap up with one more area of intense computational power. Exploiting metabolic vulnerabilities using genome-scale metabolic networks.
These networks are essentially the digital twin of a cell's metabolism. They computationally represent every single gene, protein, reaction, and metabolite interaction. There's a workflow called ER-phagy that uses a cancer cell's RNA-seq data to build one of these context-specific models.
So once you have this digital twin, how do you use it to find new drug targets?
You run in silico experiments, virtual gene knockouts. You computationally delete a gene from the network, and you observe the effect. A gene gets flagged as essential if deleting it reduces the cell's theoretical biomass production or its fitness by more than 50%.
And the key is making sure that target is cancer-specific.
Absolutely. The predicted vulnerability has to have a minimal effect on normal processes, like basic ATP maintenance. This pinpoints a cancer-specific metabolic dependency, making it a fantastic target for drug repurposing or new drug design.
This whole deep dive, from culturing patient tissue in allogeneic to these massive computational pipelines, it really underscores one central theme. The single gene, single cell line approach is dead.
It has to be. It demands an integrated, functional, and highly personalized approach. And it's also important for you to realize that drug resistance is rarely a simple switch. It often involves these complex transitory states like drug-tolerant persisters or slow-cycling cancer cells.
And you need special techniques to even find those.
Right. Things like H2B GFP pulse chase systems Studying those rare populations just reinforces the absolute necessity of these complex, multi-layered protocols we've talked about today.
Excellent synthesis. For those of you integrating this into your studies or your research, let's leave you with one final thought exercise.
Okay. If you were analyzing a new drug targeting a specific metabolic pathway in ER-positive breast cancer, why would culturing that patient tissue in alginate microstructures under continuous perfusion be vastly superior to a standard, static 2D culture using something like Matrigel?
Think about the two key conceptual failings of the older system and the two conceptual breakthroughs of the modern one. We hope this deep dive provides you with the competitive advantage you need to synthesize this incredibly complex field.